Good morning, everyone. Here I welcome you to a lecture on genetic mutations. I am not an expert in biochemistry, but I can guarantee you that I will make these genetic mutations easily understandable to you with animations and the concept categorized slides. So, without further delay, let's proceed to our topic. The first concept is missense mutation. Here, there is a mutation which will be happening in a particular nucleotide such that the entire codon sequence is altered. So what does it mean? How does this happen? So if you see here, this is how the codons are all arranged. Fine. So these three will code for methionine, and these will code for another particular amino acid, while these three will code for this amino acid, while all this will code for this amino acid. But if one particular nucleotide is changed, say for example this A is changed to G, then instead of this amino acid we will be having a totally new amino acid. So when one amino acid, when the sequence is changed, then we get a totally new protein. This is how in missense mutation one particular nucleotide is changed, and therefore that codon gets changed, and instead of the particular amino acid which it's supposed to generate, an entirely new amino acid is generated, resulting in a totally new protein. This is the missense mutation. The second one is a nonsense mutation or the premature mutation. Here, instead of forming an entirely new amino acid, that codon is changed to a stop codon. So, if it gets prematurely stopped, then what happens? It will be resulting in a protein which is also prematurely, the protein synthesis is also stopped. So therefore it will result in a truncated, often a short, non-functional protein product is the formation. Let's see its mechanism. AUG is the codon which is the starting codon. This you should know by heart. AUG stands for methionine. So in this nonsense mutation, AUG codes for methionine and AAA. So this is the second codon which codes for an amino acid 1. But instead of this A, when it gets changed to U, UAA is a stop codon. So instead of the amino acid sequence being formed and the protein being formed, the protein gets prematurely stopped because of the stop codon which is generated. What are the stop codons? UAA. So that's what we saw in our example. UAG is a second stop codon. UGA is another third stop codon. So these are the stop codons which we have to know also for sure. And if the nucleotide is changed such that any one of these is formed, then there is premature stoppage of protein synthesis, resulting in non-functional truncated protein. What do you understand by a silent mutation or wobble phenomenon? We saw in missense and the nonsense mutation that even if one nucleotide is changed, it is resulting in an entirely new amino acid. So it will be resulting in what if that is continued to happen, then it will result in an entirely new protein, and sometimes the evolution will be so fast that which is not acceptable at all. Therefore nature allows this concept of silent mutation or the wobbles phenomenon, where even though a nucleotide is changed, there will be no amino acid. I know I am confusing you. I told you previously that if the nucleotide is changed, then the amino acid also changes. But here is the catch. The alteration in the third codon position will not affect the amino acid coding. The first two nucleotides are important, but when the third nucleotide in that particular set, if that is changed, it doesn't affect the amino acid coding. So this phenomenon is known as the degeneracy or leniency of the genetic code, and this will allow for the evolutionary balance. In how does it happen? It will allow for a genetic variation, but at the same time it cannot allow all harmful mutations to happen to the human body. So therefore it is just lenient in allowing the change in the third nucleotide to happen, yet the effects are not very much pronounced. Let's see how it happens. See, this is the wobble hypothesis. So AUG codes for methionine. AUG is the starting codon which will code for the amino acid methionine. AAA codes for lysine. UUU codes for. So we have all these codons which are coding for these particular amino acids. Now this third nucleotide, that is if this is changed, still the product is lysine. The amino acid is not changed. Similarly, here also when the third nucleotide is changed, still the product remains the same, glycine. In this way, it does not allow irrational mutations to happen. or irrational protein synthesis to happen. So this is the wobble hypothesis, where there is degeneracy or leniency in the third nucleotide alone in this codon set. There is another beautiful concept that is known as a splicesite mutation, which is also known as the intron retention. In this mRNA, so mRNA is formed from DNA. So once this mRNA is formed, the mRNA has to undergo splicing. Splicing is, you need to imagine it as something which is cutting the unwanted things. So we have the intron sequence and we have exons. So exons are those which code and introns are those which are not required. So they are the non-coding. So this is our DNA, and from the DNA to mRNA we have the DNA is forming the heteronuclear RNA by RNA polymerase. The enzyme which forms a heteronuclear RNA is RNA polymerase. Heteronuclear RNA is a precursor of mRNA, where it has to undergo certain modifications to form the functional mRNA. There are three modifications. One of that is introns and exon splicing. So introns will be removed, or it will cut, or it will be thrown away, and the exons will be retained. So exons are those that are the coding part, and the introns are the non-coding part which we don't want. In this splicesite mutation, the intron exons are not cut properly. Not the exons. The introns are not spliced properly, or they are not removed properly, resulting in the retention of the intron. A common disease which happens because of the splicesite mutation is the beta thalassemia. The another good concept is frameshift mutation. Here there will be an addition or removal of a nucleotide changes the reading frame. So when one particular nucleotide is added, then it will result in the disruption of the entire reading frame. So this is how the codons are all arranged. When there is so each codon is coding for a particular amino acid, and it results in the stop codon. But now if you see, so here this nucleotide alone is removed. So what will happen? AUG codes for methionine. That means it has to started. But AAG is the one which is supposed to code lysine in this particular protein. But since one nucleotide is deleted, now we have the next codon is AGU, which will code for entirely. So this is deleted. This will code for an entirely new amino acid. Similarly, the next codon will code for another new amino acid. So finally, the final product will be entirely a totally new protein which is formed. Sometimes this can also result in premature termination, apart from producing the altered protein sequence. So that's what I'm trying to tell you. So here it is being deleted. So therefore this is producing this particular methionine, but then all these are producing entirely a new amino acids. So therefore resulting in a totally different proteins. So with this slide we have come to our end slide. I hope you enjoyed my lecture. I hope the concepts have been clear to you. So if you have any doubts, you can write to me in the comments and I will be more than happy to clear all your doubts. I have an announcement for you. I am starting my mentoring lessons for medical graduates and USMLE aspirants. If you like my slides and if you like the way I teach you, and if you want to get mentored by me, you can send a message in the comments and fill my Google form. Thank you. Looking forward to see you succeed. Have a good day.